Okay, welcome back. And let me start the slideshow. Now we're on chapter 4. We're talking about cells now. So this is really important. And so we're talking about the internal parts of cells, all type of cells, and what's called organelles, little like factories and things inside cells that do stuff. Cytoskeleton is one of the things that does that to give cells their shape. And about antibiotics are drugs that disable or kill infectious bacteria, which are cells. They're called prokaryotic cells. The idea is to give us something that won't kill our cells, that will kill the bacteria. And people couldn't even conceive of something like that before the first antibiotic, penicillin, was discovered. In fact, the guy who discovered penicillin didn't even realize, he knew it killed bacteria. And then he wrote a paper on it and then left it. And other people came to the idea that, wait a minute, if it kills bacteria, you know, we die from bacterial disease. How? Maybe we can put it into people and kill the bacteria and won't harm the person. Because before that, they used to use this thing called sulfa drugs for bacterial infections. And apparently, they were really bad, and they really did hurt and cause damage to the body. And I guess maybe they were really painful or something. I always think of the movie The Green Mile, one of our favorite movies, where Tom Hanks his character apparently had a UTI, urinary tract infection, and he was peeing razor blades. And this was in the 1930s, before antibiotics were discovered. So the only way they could be treated with these sulfa drugs, and I guess they were so bad that he'd rather pee razor blades than go to the doctor and be treated with these sulfa drugs. But he was about to go to the doctor when he was embraced by John Coffey, and miraculously, supernaturally, was cured. Anyway, so antibiotics are so precise because they bind to structures only found in bacterial cells. Like penicillin binds to an enzyme that makes their cell wall called peptidoglycan. Well, we don't have that enzyme. We don't make peptidoglycan. So antibiotics are not going to affect our cells at all. And they continue to exploit the unique structures of bacterial cells to design and discover new antibiotics. Now, some new antibiotics actually do affect our cells because they affect the way bacteria make their proteins. And we have a bacteria actually inside of our cells called the mitochondria, and it sort of affects that. But let me get on to that. Okay. Because we have to make new antibiotics all the time now because bacteria become resistant to them. If you get a bacterial infection and you go to a doctor to get an antibiotic, do they give you penicillin anymore? No, you don't get penicillin anymore. Almost every bacteria has become resistant to penicillin because they really sort of overused it. And when they first discovered antibiotics, they thought, oh, wow, we're going to defeat bacterial disease, right? Well, they didn't think about evolution. Evolution is how organisms change over time. And by changing, they can become resistant to that. Like Staphylococcus aureus, there's one form of Staphylococcus aureus that's really bad. It's a flesh-eating bacteria. And there's one strain, I think it's called MRSA, which is resistant to almost all antibiotics, and there's practically nothing you can do about it. There are some really, really seriously antibiotic-resistant bugs out there, bugs and some bacteria that can't be stopped. And that's why we need new antibiotics all the time to keep up with them and get new ones to attack them in different ways. Okay, organisms are either single celled, this is called unicellular, just as most prokaryotes, I think all prokaryotes are single celled. Protists are single cell, what are called eukaryotic cells. We're going to get into this taxonomy. Well, we talked about that taxonomy in the first chapter. We talked about the kingdom Protista. Those are called protists. Or multicellular. So unicellular is one cell. Multicellular is more than one cell, usually lots more than one cell. You are, for example, have composed of trillions of cells. And those are what are called the multicellular kingdoms, plants, animals, and most fungi. Some fungi are unicellular. The yeasts are unicellular fungi. So that's the only exception there. So I can ask you, how many cells do you need to make up an organism? Well, at least one. All right, at least one. All right. And you were unicellular at one time in your life. You were a zygote. When your dad's sperm entered your mom's oocyte, it became that single cell. So you started life as a unicellular organism, and now you're multicellular through a process called mitosis, which we're going to get to later on in the course. Okay, cell theory here. Well, the cell theory states more than just this. I don't know why the book doesn't give you all the tenets of the cell theory. First thing the cell theory states is that all living things are composed of cells. So if it's not composed of cells, it's not living. Okay. And that all cells come from earlier cells. Now that wasn't the case at one time. Other tenets of the cell theory is that cells evolved once about 3.5 billion years ago. And we know this because there are actually fossils in rocks that are 3.5 billion years old because they've been radioactively dated. And there's tiny little fossils, unicellular fossils in there. So that's where the first cells on Earth came. Those were the first cells on Earth. And so the Earth is almost about 4.5 billion years old. So it took about 1 billion years for cells to arrive, well, to either arrive or evolve on Earth. How did those first cells get there? Nobody knows. Nobody knows. And most scientists think that they got together by non-living stuff, chemicals in dilute seawater got together and formed these primitive cells. And there's some evidence about that, how electricity from lightning can make these complex molecules and have phospholipids, which make up the cell membrane. They spontaneously form these little mycelials that look like cells. So there's but nobody's been able to duplicate it in a lab, and nobody knows really how it happened. But like I said, most scientists think that cells came from non-living stuff. Now, were those first cells bacteria? They're always saying, well, the first cells on Earth were bacteria. No, they weren't. And I know that because bacteria, we call them simple cells. They're simpler than our cells, but they're actually still really complex. People used to think cells were these amorphous blobular things that weren't really complicated. Well, they are. And even bacteria are quite complicated. In fact, their metabolism is more complicated than our metabolism. So they weren't bacteria. They were some kind of really simple self-replicating nucleic acid surrounded by phospholipids. So what were they like? Nobody knows. We have fossils of them, but you can't internally look at them and see what they really were from the fossil. Now, some people think that didn't happen, that we were seeded from space. Now, you think that cells can't survive in space. Well, actually, some can. There are some bacteria that form these spores that are really hardy and can actually withstand the vacuum of space and can withstand really high temperatures. If you want a meteorite that was streaking through the atmosphere, they might actually survive. So it might actually have been seeded from other planets, but then that just puts off then cells must have evolved on some other planet. Well, how did it get there? But some people think that that was impossible. There's no way cells could have evolved from non-living things, and they think that it was supernatural, that some creator, God, made the first cells, put them on Earth, and then took a hands-off approach, and those cells evolved into all the different kinds of organs we have now. I don't know. I think more likely what's called the abiotic origin of cells. That is, abiotic means it comes from non-living material. But anyway, however they got there, they're here, all right? But if that did happen back 3.5 billion years ago, cells from non-living stuff, it can't happen today. It won't happen today. Right now, all cells come from other cells. Why is that? Because of part of the atmosphere that we're breathing in right now. We live in an oxygen atmosphere now, and there's no way molecules could self-form in like a shallow sea or something in an oxygen atmosphere. As I mentioned, I think in the essential chemistry chapter, oxygen is a really dangerous gas. It was the first, when plants first invented photosynthesis, there was the first instance of air pollution. There was no oxygen in the early atmosphere of the Earth, and fortunately there wasn't. If there had been, we wouldn't be here talking to each other, because life wouldn't have started on Earth if there was oxygen in the atmosphere. And later on, some organisms had to start protecting themselves from oxygen. They were all anaerobic bacteria, not using oxygen. And then, but when the first photosynthetic organisms started to put the oxygen in the atmosphere, they had to find a way to protect themselves. And we have special enzymes and things to protect ourselves. And also, drinking green tea, antioxidants, eating a little chocolate or coffee or bright vegetables. They are antioxidants. They help protect us against the ravages of oxygen. But these ancient organisms found a way to use this poisonous gas to make ATP, to make lots of ATP. And so one of those ancient bacteria is in every cell in your body right now. These are organelles called mitochondria. They're ancient bacteria, endosymbiotic bacteria. We'll talk more about that and the evidence for that when we start talking about organelles, because we'll mention the mitochondria. Okay, so since we live in an oxygen atmosphere, all cells come from other cells now by cell division. Types of micrographs. We don't have to worry about that. Size ranges. Okay. Cells are small. Most cells are microscopic, as a matter of fact. You can't see them with the naked eye. So here we have this range of sizes. NM is nanometer, which is a billionth of a meter. Then we have a micrometer, which I think is 1,000th of a meter, and then a millimeter, which is 100th of a meter, and so on and so forth. And we get full meters. And centimeters, which is the No, I'm sorry, I'm wrong. Centimeter is 100. It's 100 centimeters in a meter. And then it's 1,000, yeah, and so there's 1,000 millimeters in a meter. Okay. Anyway, smallest bacteria so these things are not alive. Below this 100 nm, these things are not alive, right? Including viruses. Smallest bacteria are that size, and then plant animal cells are up 100 called micrometers. So a thousandth of a, a micrometer is a thousandth of a millimeter, and a millimeter is a thousandth of a meter. And these plant animal cells, that's too small to see. And paramecium are protistans, they're a little bit larger. And some of them you can see with your naked eye, they're swimming in a pond and you can just barely see them. And actually, there's one human cell, if you have really good eyesight, you can just barely see it. It's a human egg cell. But speaking about egg cells, wait a minute. What about frog eggs and chicken eggs? Those are pretty big. But that's a little misleading. On a chicken egg, if you break open an egg, you see all the white of the egg, which is really clear, and then the yolk, right? Well, that's all non-cellular stuff. There is a tiny little microscopic cell on the yolk, on the surface of the yolk, that is the actual chicken embryo, or would become an embryo had it been fertilized. The eggs we buy in the grocery store, they haven't been fertilized. Sometimes in organic markets you can get fertilized cells. And then when you break open the egg, you can actually see a tiny little chicken embryo on the yolk. And that kind of grosses some people out, so they don't do that. But anyway, the same thing with frog eggs. Most of it's the gelatinous material, and it's a yolk, and it's a tiny little cell on the surface of the yolk. So that's kind of misleading. But sometimes the length of some cell processes can be quite long. As I said, I'm moving these fingers here. The muscles that do that are here, but the signal that's causing those muscles to contract is coming from my spinal cord. And actually from the left side of my brain, actually, if I move my right fingers, sends a signal from the brain to here, then from here all the way out to here, which is like 2/3 of a meter or something. So they can be quite macroscopic. But the process itself is microscopic. You can't see it. The nerve is not visible. You have to see it under a microscope. Okay. Why are cells small? That's something we're going to examine in the cell transport lab, or I think the water lab. We say, why are cells small? The short answer is to maximize the rate of diffusion. The smaller something is, the more rapidly things diffuse in and out of it. And so cells have to be, and cell transport has to be really fast. And therefore cells are small to make sure it is fast. We'll describe, this is called the surface area to volume ratio. We'll discuss that more in lab, actually. Okay, so we're going to get into a little bit of more classification. Biologists classify all life into three major groups called domains. The domains are the most inclusive categories, the largest categories with the largest number of organisms in it. There's three. There's domains bacteria, archaea, and eukarya. Domains bacteria and archaea are composed of prokaryotic cells. These are simpler, structurally simpler than the eukaryotic cells that are found in the kingdom Eukarya. They're also much smaller. But they are still, as I mentioned before, they're still pretty complicated because their metabolism or the chemical processes are sometimes even more complicated than eukaryote. But they don't have what's called membrane-bounded organelles. Internal membrane structures. There's no internal membranes in prokaryotic cells. So they don't have a nucleus, for example. Okay, so bacteria and archaea, they're both bacteria, actually. The archaea have bacteria that are called archaebacteria. And the difference is the bacteria, the members of the family bacteria live, where do they live? Well, everywhere. They're all over the place. Archaebacteria live in extreme environments, extreme of temperature, extreme of saltiness, or extreme pressure. And they're called the archaebacteria. And the domain eukarya includes protists, plants, animals, and fungi. And those are composed of eukaryotic cells. So table 4.1 compares their characteristics. So there we go. As I mentioned, prokaryotic cells first evolved approximately 3.5 billion years ago. Well, as I mentioned, those first cells probably wouldn't be, I think it's incorrect to call them a prokaryotic cell because they're probably simpler than that. They're probably really simple. And they weren't bacteria. They couldn't have been. I don't think these chemicals could have gotten together and made something as complicated as a bacteria. But when the prokaryotes evolved from those first cells, they were there for the first 2/3. I think they were there for the next 2 billion years was the age of bacteria. The only living thing on Earth were bacteria. And then the eukaryotic cells evolved. So that was 2.1 billion years ago. So I guess about 1.4 billion years was the length of the age of bacteria, just bacteria. Prokaryotic cells found in bacteria and archaea, those are the two domains, and eukaryotic cells found in protists, plants, fungi, animals. They're smaller, simpler in structure, not necessarily in chemistry, larger, more complex. Most have cell walls, have capsules, fimbriae, and or flagella. But the flagella are different from eukaryotic flagella. Eukaryotic flagella are actually membrane-bounded. Plant cells have cell walls, animal cells are surrounded by an extracellular matrix. Remember, animal cells don't have a cell wall. They have a plasma membrane. We have mentioned, we actually should have mentioned before prokaryotes and eukaryotes is what all cells have in common. What do all cells have in common? Well, they all have a cell membrane, right? And they all have a cytosol or cytoplasm. That the cell membrane surrounds. They all have at least one chromosome with DNA in it. So DNA is the genetic material for all life on Earth. So every cell has to have some DNA. And they have to have an organelle called a ribosome. That's not membrane bound. That the cells need to copy the information from the DNA and to make proteins, because everybody needs proteins. They have a nucleotide region containing a single circular chromosome. As I said, everybody's got to have a chromosome. And the eukaryotic cells have a nucleus containing one or more linear chromosomes. The prokaryotic chromosome is circular. And they both have ribosomes, like I said. Everybody's got to have ribosomes. So how is the nucleotide region of a prokaryotic cell different from the nucleus of a eukaryotic cell? No membrane around it. There's no membrane around the nucleoid region of a prokaryotic cell. So there's a prokaryotic cell right there, plasma membrane, cell wall, capsule, these fimbriae attaching structures, nucleoid region, ribosomes, and flagella, and some bacteria have flagella. Here's an idealized animal cell and plant cell. So you can see it's a little more complicated as far as the what are called the organelles that are found inside, that are membrane bounded. And we're going to go over in the later parts of this chapter all of these, all the important organelles. You're going to need to know the name of the organelle and basically what it does. So we're going to look at things like the nucleus, mitochondria, endoplasmic reticulate, Golgi apparatus, centrosomes, lysosomes, or you mentioned ribosomes, and in plant cells we get a different one that's called a chloroplast, and vacuoles, which are vesicles, which are little phospholipid bubbles inside cells. So we're going to go over each one of those. The idealized animal cell and the plant cell has got the same parts that the animal cell has, but with the introduction of chloroplasts, where photosynthesis takes place. Because plants do photosynthesis, most of them do. Animals do not. Also, plants have a cell wall. Animals do not. And a central vacuole, which is also not in animal cells. A vacuole is a big space that's surrounded by a membrane that usually holds some watery fluids or sometimes some nutrients. Okay, the plasma membrane, or the cell membrane, separates a living cell from its non-living surroundings. They're composed of these molecules called phospholipids, arranged into a two-layer sheet called a phospholipid bilayer. They're look like double lollipops. A head, which is actually hydrophilic, and two chains that are actually fatty acids that are hydrophobic. And because remember what the plasma membrane does, it separates two aqueous solutions, two water-based solutions from each other, so they don't mix. So what you have is the heads of the phospholipids point outside the cell, the water fluids on the outside, and they point to the inside, the water fluid on the inside. And the tails are in the inside, and it forms a hydrophobic boundary between the outside fluid and the inside fluid. So the plasma membrane is a fluid mosaic. Because molecules can freely move past one another, and it's like a phospholipid sea. And the other aspect of cell membranes is the membrane proteins. There's a whole diversity of proteins, and we'll get into later on in the chapter what they do. A lot of them are transport proteins, receptor proteins that ion channels that let things cross the membrane. So these proteins are like icebergs floating in a phospholipid sea. And there is the structure. Here are the phospholipids. As you said, hydrophilic head pointing to the outside. Hydrophilic heads here pointing to the cytoplasm inside the cell. These are both aqueous solutions. And there's the tails, the yellow, which is the hydrophobic boundary, right? That can we separate two hydrophilic solutions. If we didn't have that, then the water inside and outside would dissolve the membrane. So we can't have that happen. So, and then here are these proteins embedded in the membrane. Some of them extend all the way across, some extend part of the way across, and they do all kinds of functions that we're going to be discussing later on. So like I said, those are like icebergs in a phospholipid sea. Okay, I'm going to leave it there, and this is going to be 106 cells 1. So I'll stop it now and I'll pick it up again, so long for now.